If we are going to the already we are completed non-cyclic electron transport. Now itself we are going to the cyclic electron transport. So what are the main important salient features of the cyclic electron transport? So this cyclic electron transport takes place within the stroma lamellae. Takes place within the stroma lamellae. So why does it is not takes place within the grana? So it takes place only in the stroma lamellae. So because of lack of the enzyme, that enzyme is called NADP oxidoreductase, or also called NADH dehydrogenase, and that PS2, those are absent within the cyclic electron transport. Means it takes place in stroma lamellae. Non-cyclic does not take place within the stroma lamellae because of the within that stroma lamellae main enzyme is absent. NADP dehydrogenase enzyme is absent, and also PS2 is also absent in the stroma lamellae. It having only the cyclic electron transport takes place within the stroma lamellae. So it takes place when that additional ATP is required. So it may be plant required additional ATP, such a time it can operate in the cyclic electron transport. It is carried out with the help of only one photosystem, that is PS1. That reaction center is P700. So there is no photolysis of water. There is no photolysis of water. Photolysis of water does not take place. There is no involvement of H2O. There is no production of NADPH. So water involvement undadu, NADPH production undadu. There is no releasing of oxygen. Oxygen production kuda undadu. But what is the benefit of the cyclic electron transport? Very simple, to requirement of the additional ATP to the plant body. So it may be required additional ATP to the plant body. Such a time only that cyclic electron transport is operate within the stroma lamellae. Ee stroma lamellae so enzyme undadu, PS2 undadu. So that's why there is no non-cyclic electron transport does not take place within the stroma lamellae. Only cyclic electron transport takes place within stroma lamellae of the thylakoids. So next you can see how those electrons are moved from one electron acceptor to another electron acceptor in cyclic manner. So it is starting with PS1. PS1 reaction center P700. So that P700 is absorbing the light energy from LHC. Those are in exciting condition. So those are in exciting state. So it is undergoes to the exciting state, that is P700 star. Immediately that can be lost their electron. So that losing electron is accepting by to the another electron acceptor, that is called pyridoxine. So when it is receives the electron from to the P700, that P700 is undergoes to oxidized state. So that electron is reached to the pyridoxine. That electron will reach to the pyridoxine. So in that pyridoxine, then after that electron does not move into NADP reductase because lack of that enzyme in stroma lamellae. So these are accepting by to the another electron acceptor, that is called cytochrome B. From cytochrome B to that electron will move into quinone cycle. So simply said to be Q cycle. In that Q cycle, you can see in that Q cycle, that is plastosemiquinol is already in partial reduced condition, that accepting the electrons from to the cytochrome B and accepting 2 protons from to the stroma. So it is accepting the 2 protons from to the stroma. It is accepting 2 protons from to the stroma. It is reduced into PQH2. PQH2. When it is undergoes to oxidized, will release the energy, will release the electron, and 2 protons are translocate into the lumen. So inside this is the lumen, outside this is the stroma. ఎందుకు అంటే electron transport takes place within the stroma lamellae. Inside that stroma lamellae is called lumen. Outside the stroma lamellae is called stroma. So with the help of 1 electron, 2 protons are translocate from stroma to the lumen. 2 protons are translocate from stroma to lumen. Okay. So then after this electron is accepting by to the another electron acceptor, that is called cytochrome F. Cytochrome F2, that electron move into the another electron acceptor, that is plastocyanin. So finally that electron is reback into P700. That means PS1 reaction center is the P71. So P700. So finally that is reback into the P700. When you can see the sequence of the electron transport in cyclic electron transport starting with the PS1. That electron is moving to the pyridoxine to cytochrome B, cytochrome B to quinone cycle to cytochrome F, cytochrome F to plastocyanin, and finally reback into the PS1. Hence that electron is moved into different electron acceptors and finally reach to the PS1. Hence it is called cyclic electron transport. What is the benefit of the cyclic electron transport? So main important benefit enti ante, so in that cyclic electron transport, the main important benefit of the cyclic electron transport is accumulation of the H plus within the lumen. Every 1 electron will provide 2 protons. Every 1 electron will provide 2 protons into the lumen through the quinone cycle. Every 1 electron, if 1 electron is enter into the quinone cycle, that can translocate 2 protons, into lumen. That is main important source to ATP synthesis in becoming step. So if maybe 1 electron will translocate the 2 protons, if maybe 2 electrons translocate 4 protons, if maybe 3 electrons translocate 6 protons. You can remembering that one 3 protons are equal to the 1 ATP in the next step. 3 protons are equal to 1 ATP in next step. You know that one previously 6 protons are sufficient to production of the 2 ATP. Now itself for also 1 electron ko saripotla, 2 electrons anko okati migilipotundi. For every 3 electron will translocate 6 protons into lumen. Those are sufficient to production of the 2 ATP. So only that ATP is produced during the cyclic electron transport. So during that cyclic electron transport. So next thing finally we are going to the photophosphorylation. That photophosphorylation means formation of the ATP in the presence of sunlight. That is called photophosphorylation. That photophosphorylation is explained by next step. That is called chemi-osmotic theory, was proposed by Peter Mitchell. Next we can go to the another topic, that is called chemi-osmotic theory, and also nothing but photophosphorylation. You can go to that one. So now itself we are going to the photophosphorylation. So that photophosphorylation is aided by one of that special theory, that is called chemi-osmotic theory. that chemiosmotic theory was proposed by to the Peter Mitchell. He was also got that Nobel Prize to explain the photophosphorylation. First of all, you know the meaning of photophosphorylation. Very simple. Formation of the ATP from ADP and inorganic phosphate in the presence of light. In the presence of light, that is simply called photophosphorylation. So that photophosphorylation it takes place within the chloroplast. Photophosphorylation takes place within the chloroplast. So how that photophosphorylation takes place? So it was first discovered by Arnon. It is first discovered by Arnon. So in one of the bacteria that is called Rhodospirillum. One of the bacterium Rhodospirillum. So how that photophosphorylation is carried out? It was explained by one of the special theory. That theory is called chemiosmotic theory. That chemiosmotic theory was proposed by Peter Mitchell. So that chemiosmotic theory was proposed by to the Peter Mitchell. So according to Peter Mitchell, the main driving force to synthesis of ATP in the presence of light is the accumulation of large number of protons. Accumulation of the large number of protons within the lumen of the thylakoid or grana that can be providing the energy to synthesis of ATP. So it can be explained with the help of this diagram. So this is the grana. So this one is the grana. Inside that grana, that is lumen. So outside that lumen is simply said to be stroma. So main important driving force to synthesis of ATP is the accumulation of large number of protons within the lumen that providing the driving force to the synthesis of ATP. So first of all, you can see how those H plus ion concentration or proton concentration is increased within the lumen. So there are three important ways to increasing the H plus ion concentration. To increase the H plus ion concentration in lumen. So H plus increase within the lumen by three important events. What are those three important events? First of all, you know very well, that is the photolysis of water. So that photolysis of water takes place within the lumen. So inside that lumen, that water molecule is dissociate electrons and protons. So that water molecule dissociates into two electrons and two protons and consequently releasing the oxygen. So that photolysis of water takes place within the lumen of the thylakoids. That is also every water molecule will provide two protons directly into the lumen. That is the first event to accumulation of the protons within the lumen. And second one, the quinone cycle, already discussed in previous class. So quinone cycle of the non-cyclic electron transport. So that non-cyclic electron transport, quinone cycle are also providing protons. Also providing protons into lumen. For suppose this is the quinone cycle, that quinone also provide every two electrons or four protons are for two electrons, four protons are translocated within the lumen from to the stroma. So stroma నుంచి ప్రతి ఎలక్ట్రాన్కి రెండు ప్రోటాన్లు వస్తాయి. ప్రతి two electrons వచ్చేసి four protons. When we are observing the non-cyclic electron transport quinone cycle, every one electron will aid or translocate two protons from stroma to lumen. So this is also another important event to increasing the H plus ion concentration within the lumen. And finally, that quinone cycle. So ipude cheppukunnam quinone cycle of the cyclic electron transport. So that cyclic electron transport is also in this one quinone cycle, that quinone cycle also providing the translocate the protons from stroma to lumen. So every one electron will translocate 2 protons in quinone cycle of the cyclic electron transport. So cyclic electron transport లో కూడా మనకి quinone cycle ఉంటుంది. That quinone cycle every electron will translocate 2 protons from stroma to lumen. Anyway, overall those proton concentration is increased within the lumen. సో దీంతో పాటు ఇంకోటి కూడా జరగాలి. సో ఎప్పుడైతే ప్రోటాన్ కాన్సన్ట్రేషన్ లూమెన్లో ఇంక్రీజ్ అయిందో మరి స్ట్రోమాలో ప్రోటాన్ కాన్సన్ట్రేషన్ తగ్గిపోవాలి. హౌ ఈజ్ ఇట్ పాసిబుల్? సో దేర్ ఆర్ ఆల్సో 3 ఇంపార్టెంట్ ఈవెంట్స్ టు డిక్రీజింగ్ ద H plus concentration within the stroma. H plus decrease within the stroma. So already you know very well these two. One is non-cyclic quinone cycle, non-cyclic Q cycle, and another one cyclic Q cycle. So these two are also translocate the protons from stroma to the lumen. So ultimately the proton concentration will decrease within the stroma when compared with the lumen. And also third one, what is that third one means? Reduction of the NADP plus. That NADP plus is accepting 2 protons from to the stroma, that is reduced into NADPH. That is reduced into the NADPH. So that is also another important event to reducing the H plus concentration, to reduce the H plus concentration, to reduce the H plus concentration within the stroma. ఇప్పుడు చూస్తే మనకి లూమెన్లోనేమో H ప్లస్లు ఎక్కువ ఉన్నాయి. సో వెన్ వీఆర్ కంపేర్ విత్ లూమెన్ అండ్ స్ట్రోమా, ద కాన్సన్ట్రేషన్ ఆఫ్ ద ప్రోటాన్స్ 1000 times more within the 1000 times more within the lumen when compared with the stroma. సో స్ట్రోమాతో కంపేర్ చేసినప్పుడు లూమెన్లో ప్రోటాన్స్ చాలా ఎక్కువ ఉంటాయి. మరి ప్రోటాన్స్ ఎక్కువ ఉన్నాయనుకో, దోస్ ఆర్ ట్రై టు ఎస్కేపింగ్ ఫ్రమ్ టు ద స్ట్రోమా టు lumen. Those are escaping from stroma to lumen. But this membrane, this thylakoid membrane, are also grana membrane. That grana membrane is impermeable to translocation of the grana membrane is impermeable to translocate the protons. Grana membrane is impermeable to the translocation of the protons from lumen to stroma. But how is it possible to translocate the protons from lumen to stroma? Lumen లోనేమో protons చాలా ఎక్కువ ఉన్నాయి, స్ట్రోమాలోనేమో protons తక్కువ ఉన్నాయి. ఇప్పుడు లూమెన్లో ఉన్న protons అన్నిటి రావాలి, సో స్ట్రోమాలోకి రావాలి. ఇది ఎలా పాసిబుల్ అండి? ఇన్ దట్ థైలాకాయిడ్ మెంబ్రేన్ ఆర్ గ్రానా మెంబ్రేన్ there is one of the enzymatic protein or enzymatic complex. That enzymatic complex is called ATP synthase. ATP synthase are also called F0 F1 particle. It is also called F0 F1 particles. ఇంకొక పేరు కూడా పెట్టొచ్చు. It is also CF0 CF1 particle. So CF0 CF1 అంటే coupling factor. So it is also called coupling factor, are also said to be. So nowadays ఈ వర్డ్ వాడుతున్నాం ఎక్కువగా, అదేంటంటే F0 F1 particle. So F0, that F0 is embedded within the thylakoid membrane. That F0 is a stalk piece or tail piece. That stalk piece is called F0. So that F0 is embedded within the thylakoid membrane. So membrane లోపలికి F0 ఉంటుంది. Okay, but what is the benefit of the F0? It is helpful to the translocation of the protons. Translocation of the protons from lumen to the stroma. Those are mainly helpful to the translocation of the protons from lumen to the stroma. So that is also So that is also mainly helpful to the with the help of F0. But what is the function of this headpiece? That headpiece is the F1 that is embedded within the stroma. ఈ headpiece వచ్చేసి స్ట్రోమా లోపలికి ఉంటుంది. మరి what is the function of that F1 particle? So that F1 particle is mainly helpful to the synthesis of ATP, adenosine triphosphate. That is the energy currency in the cell. But how is it possible? So through the F0, if every 3H plus can be moved from lumen to the stroma, that can produce large amount of the energy. Book lo iవ్వలేదు కానీ nearly 13.7 kilocalories energy is released. So that energy is useful to the making of or preparation of the 1 ATP molecule. So that energy can be utilizing by ADP, adenosine diphosphate. In the presence of inorganic phosphate, it can be converting into ATP. That means every 3H plus equal to 1 ATP. Every 3H plus equal to 1 ATP. So when that 3H plus or 3 protons are across the membrane from lumen to the stroma, that can be released the large amount of energy. That energy is useful to the making of ATP. That ADP is combined with inorganic phosphate to form the ATP in the presence of light. Hence it is called photophosphorylation. It was explained by chemiosmotic theory. That chemiosmotic theory was proposed by Peter Mitchell. According to Peter Mitchell chemiosmotic theory, the main important driving force to synthesis of ATP is the accumulation of large number of H plus ions within the lumen that produce every 3 protons are released. When that every 3 protons are translocated from lumen to stroma, will release the energy. That energy is useful to synthesis of 1 ATP. So this is the chemiosmotic theory. That chemiosmotic theory is mainly helpful to the synthesis of ATP. So that ATP synthesis takes place with the help of chemiosmotic theory. Okay. So you can see what are the end products of light reaction. So finally you can see the end products of light reaction. Already in non-cyclic electron transport, NADPH and also ఇక్కడ చూస్తే మనకి ఏం ఫామ్ అయింది ATP. దాంతోపాటు నాన్ సైక్లిక్లో మనకి ఏమొచ్చిందండి, ఇన్ దట్ నాన్ సైక్లిక్ ఆక్సిజన్ ఈజ్ ఆల్సో రిలీజ్. సో దీస్ 3 are considered as a end product of the light reaction. So 2018లో ఒక నీట్ బిట్ ఇచ్చాడు. The following is not formed in the light reaction of the photosynthesis అన్నాడు. అప్పుడు వాడేమి ఇచ్చాడంటే NADP, ATP, O2, దాంతోపాటు ఇంకోటి ఇచ్చాడు. అదేంటి అంటే NADH. This NADH is also another coenzyme. So NADH is also another coenzyme, but it is involved into the respiratory pathway, but not in photosynthesis. So that's why what are the end products of the light reaction means? So NADH and ATP. So both are considered as assimilatory power or assimilation energy. So ATP and NADPH are considered as a assimilatory power or assimilation energy. Okay, so this is the light reaction. So in that light reaction finally ఈ ఆక్సిజన్ బయటికి వెళ్ళిపోతుంది. ఇంకా మిగిలింది ఎవరయ్యా అంటే NADPH and ATP. That is the assimilatory power. So already light reaction is completed in previous one. So now itself we can go to the dark reaction. So the dark reaction takes place within the stroma. So it is dark reaction is also called carbon assimilation or carbon fixation. carbon assimilation or carbon fixation. It takes place within the stroma of the chloroplast. What happened during the dark reaction? That carbon dioxide undergoes to reduction and producing into carbohydrates, and produces the carbohydrates by utilizing the energy that was synthesized during the light reaction. That is called assimilation energy or assimilatory power, ATP and NADPH. So that ATP and NADPH, that assimilatory power was synthesized during the light reaction, that is utilized within the dark reaction. So in the dark reaction was first identified by three scientists, Melvin Calvin, Benson and Basham. But in the honor of the Calvin, this cycle is called Calvin cycle. They were worked on one of the unicellular alga. That alga is called chlorella. So that unicellular alga is said to be chlorella. Unicellular alga, they were utilizing the radioisotope C14. Actually C12 kada, but C14 radioisotope is used to that radioisotope is used to synthesis of the carbon or maybe what is this one, glucose or carbohydrates. So the type of Calvin cycle is also called C3 cycle. How can you say that C3 cycle? So the first step in Calvin cycle, so that carbon dioxide is accepting by to the one of the pi carbon containing compound, that is called RUBP, to form into a phosphoglyceric acid. So that phosphoglyceric acid having three carbons, hence it is called C3 cycle. That Calvin cycle is also called C3 cycle because that first formed compound is the phosphoglyceric acid. That phosphoglyceric acid containing three carbons, hence it is called C3 cycle. So next you can go to how that Calvin cycle. So the primary acceptor of the CO2 in Calvin cycle is the ribulose 1,5-bisphosphate. So that is the pentose sugar, 5 carbon containing sugar, ribulose 1,5-bisphosphate. So in this one, that is the primary acceptor of the CO2. So that primary acceptor of the CO2 and first formed compound is the phosphoglyceric acid. That phosphoglyceric acid containing 3 carbons, hence it is called C3 cycle. That is called C3 cycle. You can go to that flow chart with that C3 cycle containing 3 important reactions. C3 cycle having 3 reactions. So there are 3 steps in the Calvin cycle. So one is called carboxylation, second one is the reduction, and the third one is the regeneration. In that first step, that PCA is formed. In that next step, that PCA is converting into G3P, and finally RUBP is regenerated, will producing into glucose molecule. It can be explained with the help of one of the big flow chart. So with the help of that flow chart, you can be see the Calvin cycle. For our convenience purpose, you can go to the flow chart with 6 molecules. So that 6 molecules of the RUBP will combined with 6 molecules of the CO2. 6 molecules of the CO2 will form into 12 molecules of phosphoglyceric acid. So in the presence of enzyme Rubisco, this is called carboxylation. This reaction is called carboxylation. So this is the first step in Calvin cycle. That RUBP is accepting the 6 molecules of carbon dioxide. That RUBP means ribulose 1,5-bisphosphate. Ribulose 1,5-bisphosphate. It is a pentose sugar. So that is accepting the CO2 and producing into 12 molecules of 2-phosphoglyceric acid. 12 molecules of the 2-phosphoglyceric acid in the presence of enzyme Rubisco. Ribulose 1,5-bisphosphate carboxylase oxygenase. It is the most abundant protein in the world or in the plant kingdom. That is the Rubisco. So this is the carboxylation. In that next step, that 12 molecules of the 2-PCA, 3-phosphoglyceric acid, 12 molecules of the 3-phosphoglyceric acid, that can be utilized the 12 molecules of ATP. So that is converting into 12 ADP. So in the presence of enzyme kinase, it can be producing into 12 molecules of 1,3-bisphosphoglyceric acid. No change in their carbon number. So if maybe ATP is involved, enzyme is called kinase. In that next step, in the reduction, so 1,3-bisphosphoglyceric acid, by utilizing the 12 molecules of NADPH that was synthesized during the light reaction, and hydrogens తీసుకుంటుందమ్మా. So that is converting into 12 molecules of NADP plus will producing into 12 molecules of G3P. G3P means glyceraldehyde 3-phosphate. It is also called PGAL, phosphoglyceraldehyde. 3 molecules of the PGAL. PGAL means phosphoglyceraldehyde. So in this one, that G3P, for our convenient purpose, it can be converts into the 6 into 2, 12 molecules, G3P pool. So this is the G3P pool. For our convenient purpose, it can be converting into 6 into 2. And 12 molecules, 1, 2, 3, 4, G3P. So everyone is 2 molecules. Everyone are the 2 molecules. So those 2 G3P molecules are enter into the cytoplasm. Those 2 G3P molecules are enter into the cytoplasm, will producing into glucose. Will produce the glucose. You can see this one. Every G3P having 3 carbons. Every G3P containing 3 carbons in the presence of enzyme dehydrogenase. So that is enter into the cytoplasm and converting into glucose, C6H12O6. 2 molecules of the G3P. 2 molecules of the G3P enter into the cytoplasm and producing into glucose molecule. So next, that remaining 10 molecules of the, out of 5/6% of the G3P, glyceraldehyde 3-phosphate is useful to regeneration of the primary product, ribulose 1,5-bisphosphate. So it is the third step. So in that regeneration, so that G3P is converting into DHAP. G3P is converting into DHAP. That G3P and DHAP are the isomers. So 3 carbons, 2 molecules. Those 2 molecules of the G3P is converting into DHAP in the presence of enzyme isomerase, triose phosphate isomerase. triose, phosphate, isomerase. It is converting into DHAP. In that next step, the DHAP is combined with another G3P molecule. So DHAP combined with another G3P molecule will producing into fructose 1,6 bisphosphate. 6 carbons. 3 carbons from DHAP, 3 carbons from G3P. Total 6 carbons. So molecules 2. Okay. Fructose 1,6 bisphosphate in the presence of enzyme aldolase. In the presence of enzyme aldolase, that is converting into fructose 1,6 bisphosphate. Fructose 1,6 bisphosphate ante 2 phosphates are attached to the first carbon atom and the 6th carbon atom of the fructose. Remembering that one. So ఇప్పుడు fructose 1,6 bisphosphate లో ఈ ఫస్ట్ కార్బన్ దగ్గర ఉన్న ఫాస్ఫేట్ లాస్ అవుతుందిరా. Hence it is converting into fructose 6 phosphate. Hence it is converts into fructose 6 phosphate. One phosphate can lose from to the first position in the presence of enzyme phosphatase. In the presence of enzyme phosphatase. Next, we'll come to the G3P. Another 2 molecules of the G3P. Those are combined with fructose 6 phosphate. So ఇక్కడ 6 కార్బన్స్ ఉంటాయమ్మా, ఇక్కడేమో 3 కార్బన్స్ ఉంటాయి. 3 కార్బన్స్ 6 కార్బన్స్, ఇప్పుడు టోటల్ 9 కార్బన్స్ కదా. those are combined with fructose 6 phosphate is combined with another G3P, will producing into 2 compounds, will dissociate it into 2 compounds. One is called erythrose 4 phosphate, 4 carbon containing compound. So 2 molecules same ra. Another one is the xylulose 5 phosphate. It also having 5 carbons. Xylulose 5 phosphate, that is also 5 carbon containing compound in the presence of enzyme ketolase, trans ketolase. It is converting into 5 carbon containing compound. So that is the xylulose 5 phosphate, erythrose 4 phosphate. So both are combined to form into 9. 6 plus 3 9, 4 plus 5 9. It is also 2 molecules. So next come to the another G3P. Another 2 molecules of the G3P will converting into DHAP, will combine with erythrose 4 phosphate, will combine with erythrose 4 phosphate in the presence of enzyme aldolase. So it is converting into 4 carbons, 3 carbons, total 7 carbons. It producing into 7 carbon containing pseudoheptylose 1,7 bisphosphate. It form into pseudoheptylose 1,7 bisphosphate. That can be produced the pseudoheptylose 1,7 bisphosphate. It having 7 carbons, 2 molecules, no chain. Okay, in the presence of enzyme trans aldolase. చూడండి ఇక్కడ ఆల్డోలేజ్ డిహెచ్ఏపి ఇన్వాల్వ్ ఉందంటే ఆల్డోలేజ్, ఇక్కడికి వచ్చేలోపు కీటోలేజ్, మరలా ఏమైంది డిహెచ్ఏపి ఇన్వాల్వ్మెంట్ ఉంది ఆల్డోలేజ్. సో దట్ pseudoheptylose 7 phosphate అంటే 2 phosphates, 1 phosphate is attached to the first carbon atom and another phosphate is attached to the 7th carbon atom. Hence it is pseudoheptylose 1,7 bisphosphate with 2 molecules. And also it can be lost there, phosphate from to the first carbon, it converting into pseudoheptylose 7 phosphate. It converts into the pseudoheptylose 7 phosphate. So no change in their 2 molecules. Next, finally, last G3P is present. Final G3P. That final G3P is also combined with pseudoheptylose 7 phosphate. That G3P having 3 carbons. So that G3P having the 3 carbons with the 2 molecules. So that is combined with the pseudoheptylose 7 phosphate. It having 7 carbons. So 7 plus 3, 10 carbons. it can be dissociated into xylulose 5-phosphate and ribose 5-phosphate, xylulose 5-phosphate, xylulose 5-phosphate and ribose 5-phosphate. It having 5 carbons, it also having 5 carbons, no change in their number. So these are also true. So 2 molecules of the ribose 5-phosphate. So those 2 molecules of the ribose 5-phosphate, these 2 molecules of the ribose 5-phosphate is converting into ribulose 5-phosphate. No change in their carbons, 2 molecules. Okay, so no change in their carbons in the presence of enzyme isomerase. ఇక్కడ మనకి ఆల్డోలేజ్ వచ్చింది కదా, ఇక్కడ ఎంజైమ్ కీటోలేజ్, ఎంజైమ్ వచ్చేసి కీటోలేజ్. ఇప్పుడు ఏమవుతుందండి, ఇన్ ది ప్రెసెన్స్ ఆఫ్ ఎంజైమ్ ఐసోమరేజ్ దట్ ribose 5-phosphate is converting into ribulose 5-phosphate. And also you can see this one, 2 molecules of the xylulose phosphate is also converting into ribulose 5-phosphate. Xylulose 5-phosphate is also converting into ribulose 5-phosphate. This xylulose 5-phosphate are also converts into ribulose 5-phosphate. Totally ఇవి 2 ఇవి 2 ఇవి 2 మొత్తము total 6 molecules of the ribulose 5-phosphate is formed. కానీ మనకు కావాల్సిందేంటి, ribulose 1,5-bisphosphate, but now itself it available only 2 minutes. So now itself it is only ribulose 5-phosphate. Finally that ribulose 5-phosphate, by using the 6 molecules of ATP, by using 6 molecules of the ATP that converting into 6 molecules of ADP and a phosphate తీసేసుకుంటుందిరా in the presence of enzyme kinase, and finally producing into regeneration of the 6 molecules of ribulose 1,5-bisphosphate. Finally produces the 6 molecules of the ribulose 1,5-bisphosphate. It is also primary acceptor of the CO2. ఎంత ఎక్కువ ribulose 1,5-bisphosphate ఉంటే అంత ఎక్కువగా CO2 తీసుకుంటుంది, అంత ఎక్కువగా మనకి గ్లూకోజ్ ఫామ్ అవుతూ ఉంటుంది. సో ఇప్పుడు చూడండి, ఇక్కడ 6 కార్బన్స్ ఇన్వాల్వ్ అయినాయి అనుకోండి, మనకి ఒక్క గ్లూకోజ్ వచ్చింది. మరి how many ATP are utilized? You can see this one, 12 ATP and also 6 ATP for 1 glucose formation. For 1 glucose, how many ATP are required? 12 plus 6, total 18 ATP. How many NADPH2 are produced during this process? Only 12 NADPH. Assimilatory power is required to synthesis of the 1 glucose molecule during the dark reaction or Calvin cycle or C3 cycle. మరి 1 కార్బన్ అడిగారు అనుకోండి, మేబీ ఇట్ ఈస్ ఆస్క్డ్ విత్ ఇన్ ద 1 కార్బన్. మరి 1 కార్బన్ అనగానే ఫర్ 1 కార్బన్, సో ఒక గ్లూకోజ్ 6 కదా, 18/6. సో 1 కార్బన్ కన్నాం అనుకోండి, 3 ATP and 2 NADPH. Assimilatory power is required to synthesis of the 1 glucose molecule during the Calvin cycle or C3 cycle. First formed compound is the phosphoglyceric acid. The primary acceptor of CO2 is ribulose 1,5-bisphosphate. So for 1 glucose molecule in Calvin cycle, 18 ATP and 12 NADPH. Assimilatory power is required with the help of 3 reactions: carboxylation, reduction and regeneration. Carboxylation, formation of the 3-PCA, reduction, so from to the 3-PCA, G3P, and finally regeneration of the RUBP. So that is the end products of the Calvin cycle. Thank you very much.